Thank you. Thank you very much. Thank you very much for attending. It's really a pleasure to be here today and then see so many names of friends. Okay, so our intention today, Juliana's and my intention today, is to show you the work that we are doing related to Work Package 118 regarding the emergence of prebiotic chemistry in space. So as Stephen said, so I will be presenting a first talk, which focuses basically on the work that we've been doing recently at the Center of Astrobiology regarding the detection of prebiotic molecules in the interstellar medium. And then Juliana will tell you the research that she's currently doing on trying to understand the level of chemical complexity that we can find in molecular clouds affected by the interaction of supernova remnants, and how she wants to link that to the origin of our own solar system. Okay, so our work package is 118, and that's the title that we have given it, the emergence of prebiotic chemistry in interstellar space. It is coordinated by Juliana and myself, and here I list the people who are involved in this working group. And being here at the Center for Astrobiology, in my group, we are mainly interested in trying to understand whether we can connect the chemistry that we see in the interstellar medium with basically the first steps towards the formation of the first biomolecules that led to the origin of life. So what we currently know is that life emerged about 3.5 billion years ago from simple molecular compounds. And this is what it is shown here in this diagram, where especially here at the bottom of this slide, life probably emerged from these small molecules that started reacting among themselves, forming larger and larger molecular compounds. And then at some point, what we know is that these these precursors of biomolecules started forming the first metabolic, replicating, and membrane-bounded subsystems, which at some point they started working together to form LUCA, what we call the last universal common ancestor, which all the living organisms come from. So what we currently believe is that a significant fraction of those organic compounds, of those molecules, were formed endogenously on the surface of primitive Earth. However, it is also recognized that a significant fraction of those compounds may have come from outer space. So they could have been formed exogenously, and they were delivered to the surface of Earth during the late heavy bombardment period on board comets, meteorites, asteroids, and interplanetary dust particles. So it has been estimated that between 10 to the 16th to 10 to the 18th kilograms of exogenous organic matter was delivered to the surface of Earth, which is orders of magnitude higher than what it is currently present in the biosphere. In addition, so what it has been shown in recent space missions is that amino acids, sugars, and nucleobases are found in these outer space objects. And something that has been really transformational in the past few years, and the past year, has been the results of the missions that traveled to Ryugu, to the asteroids Ryugu and Bennu, which showed that they contain the 5 nucleobases and many of the proteogenic amino acids, or the amino acids that we find in life. So also something that has become more and more evident, and especially also thanks to the Rosetta mission, is that some of those organic compounds may form already during the initial conditions of solar system formation, and in particular in the molecular cloud that led to the solar system. So what we are interested in at the moment here at the Center of Astrobiology is to try to understand how those prebiotic molecules can form during these initial conditions in molecular clouds. And this is the goal of this new project, this ERC project called the Onset of Prebiotic Chemistry in Space, which aims at not only characterizing the inventory of prebiotic molecules in the interstellar medium, but also how these molecules are inherited or are transferred throughout the process of the solar system formation, and also which tries to explore or to understand the origin of chemical complexity from using not only the traditional ways in astrochemistry, but also using novel methods based on complex network theory. Today, however, I'm going to show you the work that we've been doing regarding the inventory of prebiotic molecules in space and the work that we've been doing in the past few years to try to detect these molecules, in particular toward one molecular cloud located in the Galactic Center. So this is what I will focus on during the first part of this short talk about the work that we've been doing in the past 5-6 years here at CAB regarding the detection of these prebiotic molecules. And then I will show you how AtLAST can help us in enlarging the number of molecules of this type detected in space. Okay, so the central, you're probably familiar with this, the central molecular zone covers the inner 500 parsecs of the Milky Way and basically hosts more than 80% of the dense gas in the galaxy. Something that it was found in the early 2000s is that the GMCs, or the molecular clouds located in the central molecular zone, are very rich in complex organic molecules. So they show a very wide variety of these complex organics, and they present similar abundances across the whole CM set. In the past few years, so we have been focusing on this particular cloud, which is associated with the Sagittarius V2 molecular cloud complex. It's called G0693 minus 0.027, which is located here. So is this cross here. This is a quiescent cloud. There's no signs of a star formation undergoing in this cloud, but it presents, so what we think is that the high level of chemical complexity that is found in this cloud is associated or is produced by a large-scale cloud-cloud collision. The physical properties of the cloud, so you can see them there. The densities are of the order of a few times 10 to the 4 particles per cubic centimeter. The dust and gas are thermally decoupled, so the dust temperatures are lower than 20 Kelvin, while the gas temperatures are larger than 100 Kelvin. But the peculiarity of this cloud, the molecular emission towards this cloud, is that the excitation temperature, especially for these large molecules, are relatively low. So they present excitation temperatures lower than 15 Kelvin. And this really represents a huge advantage for the detection of new complex organic molecules, especially in crowded spectral surveys. This is illustrated here in these two panels. So in the upper panel is the prediction for, or the spectrum predicted for glycine for what we would basically detect towards a hot molecular core with excitation temperatures of the order of 100, from 100 to 300 Kelvin. As you can see, so the spectrum clearly peaks at higher frequencies. While, however, if we target objects whose physical conditions enable the excitation temperatures of the order of 10 Kelvin, 10, 15 Kelvin, where you can see that the spectrum of these large complex organic molecules shift towards lower frequencies, which are cleaner from transitions from smaller species. And also, this reduces significantly the level of line blending and line confusion. So these low frequencies really represent the sweet spot in which we can target these large complex organic molecules. And this is what we have been using, or the methodology that we've been using in the past years to detect many of these molecules. Okay, now, so what kind of molecules are we interested in? So by talking to biochemists here at the Center for Astrobiology, they pointed out this beautiful work regarding the primordial RNA wall, where they, especially from the group of Sutherland in Cambridge and now Pauner at UCL, where by starting from very simple molecular compounds, from very small chemical compounds, they can basically build up all this chemical scheme in which they can produce RNA precursors. In this particular case, they were able to show the synthesis of pyrimidine ribonucleotides, which are the building blocks, one of the types of the building blocks of RNA. They could also build amino acids, sugars, and lipid precursors. So if one inspects this diagram closely, So what one realizes is that these molecules that are with blue circles, and this was back into 2018 when I started here at CAP. So what one realizes that these molecules with the blue circles were the ones that were routinely reported back then in 2018. And then we also found some species that were also present in this chemical scheme, that are the ones in red circles, that were also very important in the synthesis of these building blocks of RNA. So some examples are this molecule, glycolonatrial, and this one, urea. So we targeted these 5 species. This is 2-aminoxazole, this is glyceraldehyde, and the hydroxyacetone using our first surveys towards this molecular cloud in the Galactic Center. We reported these 2, glycolonatrial and urea, and then encouraged by this methodology, when we decided to follow a similar procedure for detecting more prebiotic molecules. So since 2019, so we have detected, this is just a sample of the molecules that we have reported so far towards this molecular cloud in the Galactic Center. So far, we have reported 28 molecules, and most of them, or the majority of them, can be considered as prebiotic because they are related to these chemical schemes of prebiotic chemistry, which are basically trying to explain the formation of the first biomolecules. So here you can see precursors, not only of ribonucleotides, but also precursors of sugars, precursors of amino acids, and also molecules involved in the synthesis of the first protoproteins and protolipids. Okay, something that has been quite difficult for us regarding the type of prebiotic, or one type of prebiotic molecules that we have not been able to obtain any detection, are sugars. So here you can see that sugars are actually very important for prebiotic chemistry because they represent essential ingredients for the formation of ribonucleotides and, of course, of nucleobases. In particular, in these prebiotic chemistry schemes, as one can see, is that they have to be introduced also in an ad hoc manner, so as inputs in their chemical schemes, because they are very difficult to be synthesized under prebiotic conditions in the lab. So that's why we think that sugars are prime targets for our searches, because it can provide some additional input, or an additional resource for sugars in early Earth, because they could have come from outer space. Unfortunately, sugars with 3 and 4 carbon atoms have not been detected so far. So what can AtLAST do for us? And this is something that we presented recently in the white paper that we submitted in December. And this is the feasibility study for the detection of, in this particular case, so we focused on these 2 types of sugars with 4 and 5 carbon atoms. This is erythrulose, which is the only ketose sugar. So there are ketoses and aldoses, and also ribose. So this is the predicted spectrum for these 2 species. So what we estimated is that basically we require sensitivity of the order of 0.1 millikelvin, which is 10 times better than what we have in our current surveys performed with the IRAM 30-meter and the Jeves 40-meter telescopes. And for the detection of ribose with a signal-to-noise ratio above 3, and this requires an amount of observing time of 100 hours, at less than 100 hours. So if we just think about current facilities, to achieve a similar sensitivity would require longer than 1 year. So this is for ribose, but as you can see, for erythrulose, I just did a quick calculation with the sensitivity calculator half an hour ago. So erythrulose is expected to be much brighter. So its detection, its 4 or 5 signal detection will require just 20 hours. So this illustrates the power of AtLAST for the detection of these prebiotic molecules in space. In this white paper, we also indicated the type of instruments that would be useful for this kind of searches. We believe that a heterodyne multi-beam receiver would be perfect for us. It should work in this low frequency regime between 30 and 50 gigahertz, because this is the frequency regime where all these large complex organic molecules peak for these excitation conditions. This emission is actually extended and is very, very extended across the Sagittarius B2 molecular cloud. So if we have a large field of view with an angular resolution or a beam of the order of 27, 38 arc seconds, that's perfect for us. And then it depends on if the 10 millimeter array is sparse. We could do staring or wobbler observations, so where when one beam would be focusing or would be pointed at the on position, and then we would have the other beams pointing at off positions. But if it's filled tightly, we could do fast on the fly, or we could use a daisy pattern. And this is it from my side. So if you have any questions, I'll be happy to answer.